afternoon, everybody. Yes, it's Christmas, and so since 5 to 7, I've been in a media circus. Anyway, I want to tell you a little bit about 2 decades' work on Svalbard. I'm going to begin with my acknowledgements because not only are there 6 co-authors of this paper, but an awful lot of other people who have been involved over the years with different expertises, and also we've had a tremendous amount of funding over this period too. So only last month, a paper appeared in Science, a review called The Broad Footprint of Climate Change, From Genes to Biomes to People. And this diagram appeared, which documents across taxa what sort of changes we're seeing. And in particular, people have recorded morphological changes and tends to be a decline in size with climate change. People have also noticed changes in dynamics and productivity, etc., and I'll touch on all of these. So, for example, a decline in size has been recorded in Soe sheep, work that was done, led by Tim Coulson, back 6 or 7 years ago. So our study site then is in the Arctic. Rangifer, that's both reindeer and caribou, have a circumpolar distribution. We're working on Svalbard, which is some 1,000 kilometers north of the Norwegian coast, halfway to the pole. And it's an extraordinary environment to live in because, as this diagram shows, snow lasts for most of the year. There's a very short growing season and plants quickly senesce. So summer is incredibly short. During this hot winter, which is also characterized by more than 3 months of polar night, females are gestating. So in the toughest time of the year, they're pregnant. This is what it looks like. Reindeer have to dig through snow, which they can do when it's powdery, to find food, and that's all you're likely to find. The biomass is very low, and even in summer, it's not exactly luxuriant. And in fact, it is incredibly temperature sensitive. A 2-degree here change in July temperature leading to a sort of doubling in plant biomass on one of these communities, the moist heath in this case. Also, you'll be aware that the Arctic is probably warming faster than anywhere else on Earth. And in our study, this is the sort of changes that we've seen. About a 1.5 degree over those 2 decades increase in July temperature from just about 6 to 7.5. But in winter, an even more dramatic change. This multiplies up to about 9 degrees over those 20-odd years. So things are changing fast. And as you can see, the variance is very high. So in this winter, the mean January temperature was only about -3. The other significant thing is that in these periods of occasional winter warming, so what this diagram, sorry, I'm not controlling the, what this diagram shows is a period between late December 2015 and early January 2016, so New Year's here somewhere. And for a period of 6 days, temperatures were above 0. In fact, the minimum never, this is the mean, the minimum never went below 0 during that time. When temperatures are low, like -15, there's no precipitation. It's too dry. And as it warms up, you'll have snowfall, in this case, when it's about sort of -1 or -2. And then afterwards, with these high temperatures, if there is going to be precipitation, then you'll tend to find it's rain. And that's a very, very significant amount of rain falling in a concentrated period. And then as it cools down again, we go back to snow, and eventually the temperatures are cool enough that we're not going to get precipitation. So this rain on snow is actually increasing over time. I mean, it's quite stochastic, as you can see in this diagram. Here in the 95-6, I think it was, we had a very high sort of rainy winter, but generally it's tending to increase in the latter half of the study. So higher rain on snow we know leads to more ground ice because we dig 128 snow pits in the same place each year. We've been doing this since we realized how important this phenomenon was in the last 6 years. And the more rain on snow we have, then the thicker the ground ice. So sometimes it will form crust, sometimes it forms layers in the snowpack, and sometimes it will percolate right down and freeze at the ground. And at the same time, we've also got the population increasing. So it's an incredibly dynamic place at the moment with the climate warming and the population increasing. So this is an individual-based study. We've developed a method of reindeer trawling. These animals don't go around in herds, they're in small groups. We single out an animal that's of interest and we trawl it in this net between two snow scooters. We're focused on females. We've got more than nearly 4,000 captures, mainly April, May, but also in February for a 5-year period. A lot of different females. About 2/3 of them were marked as calves at 10 months of age. So we've got known cohorts. We've got 17 cohorts that have gone through to adulthood. They mature at about sort of 5 or 6. And each winter when we come back, we capture about 105 of those now adult animals, including about sort of 30-odd calves. So 17% of these animals have been caught 6 or more times. The maximum is 11 years, and they live 16. So we're getting individual data through individuals' lifetime. These are the sorts of long-term changes we've seen. In October, the animals have tended to increase in weight. We were culling a few for scientific purposes up to 2007, and that's a significant increase in the October weight, probably because of the improved productivity. In winter, the April masses are much more variable. The importance of this October mass is that it influences the probability of being pregnant. Regardless of your age, it's really driven by the mass you are at that time. Whereas in the winter, what we find is that this is dictating reproductive loss. So when animals are light, as they are in some winters, then they are likely to suffer a high loss of embryos, fetuses. Now, the animals grow quite rapidly. Body mass asymptotes at about 4 to 5 years of age. We first caught them at 10 months, and this is the sort of typical average trajectory. Skeletally, it asymptotes even earlier. So by about 2 years of age, they're at mature size. There's a slight increase there, but that's because of viability selection. What we find is that when we look at these cohorts, so all the animals born in the same year, when we look at them in terms of their mature size, we find that there's a significant variation. And we've got this sort of index of early environmental quality, which is actually based around the conditions their mothers were in, effectively, when they were in utero. So for each cohort in that right-hand plot, we're recording the average weight of the females in the April that they were in utero, but we're measuring their skeletal size as adults. And this is influenced by density and rain on snow. Now, another way to look at this then is the cumulative effect on these birth years, the cohorts. So the earliest cohort we've got is 1994 here, and they were quite heavy. And over the 17 cohorts that are plotted here, you can see there's a substantial decline. And what's quite interesting then is not only have we got a long-term decline, but we've got some years which they're particularly low. These are the years, the red ones here, are the years when the calves were in utero. And remember, this is their adult weight, but this is when they were in utero, and the red dots are the conditions when there was excessive rain on snow. There was more, the top sort of third of the rain on snow measures. Interestingly, the year before are the animals that experienced it as calves. So this had a high rain on snow in the winter that it was a calf. This has a high rain on snow in the year it was in utero. So there's been selection. We got rid of the small ones. We've only got the large ones surviving if they experienced it in the first winter. So let's have a look at that in a bit more detail in terms of, here's the overall doubling that we've witnessed in the 20-odd years. And that's the effect adjusting for the rain on snow. So I've fitted two variables, population size and the rain on snow measure in the year that these adults would have been in utero. And you can see that although there's quite a lot of variation, there is a significant decline there. If we again look at the over time increase in rain on snow and look at the contribution to those mature cohort weights, then we find again a negative relationship. And that would be true even with the high leverage of this point. Together, the density and rain on snow accounts for about 53% of the variance in those mature weights, differing between the birth years. Interestingly, this is plots of annual weights over our study. So this is actually over all 22 years, just taking the annual adult weight. So each point is a different year. We find that around 50 kilos, that the population, if it's below the average weight of females below 50 kilos, then we get population decline. If it's above 50 kilos, we get population growth. Interestingly, when we then go back to the graph you saw earlier of this sort of trend in mature body mass of the cohorts, what we find is that 50 kilos is there, and we're getting an increasing proportion of adult cohorts being lighter than that. So the answer then is, will 2 reindeer be sufficient for Santa's sleigh in the future? Thank you very much. There is a significant relationship between offspring size and maternal size. So on average, as they're getting mothers are getting smaller, then offspring are getting smaller. So there's a heritability of size.
So that was very interesting. I was wondering about the cost of lactation, and because they're lactating during the summer, and whether there was any effect that you could see on changing. I mean, the temperature wasn't changing too much in the summer, but sudden freezes and the cost of lactation.
There is the cost of lactation. So females who don't lactate because they lost their calf or didn't get pregnant enter the autumn 3 or 4 kilos heavier than females who've lactated. And relatively, the cost of lactation, as you probably know in mammals, is much greater than the cost of growing the fetus. But it looks as if these animals ditched their fetus to save their own lives.
Are you seeing similar rain on snow effects in other parts of the world?
Well, I think it's very much the oceanic part of the Arctic. I mean, after all, the Arctic is an ocean surrounded by land. So we see similar rain on snow icing events in the Canadian Arctic Islands, particularly where a very similar small caribou, the Peary caribou, and also musk oxen live. And there are these occasional quite significant die-offs. Rather sadly for those populations, there isn't this sort of annual data and no following of individuals, and you need to be able to sort of have regular information. Thank you.
Thank you. I feel a bit inadequate after 20 years of phenomenal work on reindeer, and now I'm going to talk about modelling animals having sex. Anyway, hopefully you'll just hang on for the next 15 minutes and then it'll be back to something a bit more useful. Trying to work out which one is this. So what I'm going to do in this talk is, firstly, I'm going to run us through some ideas about how sexual selection might affect adaptation, and what we're particularly interested in here, given the session, is adaptation to continually changing environments. Then I'm going to talk about some of the evidence that sexual selection actually does affect things like adaptation to changing environments and extinction rates. And then I'm hopefully going to try and pull some rather disparate threads together using an individual-based eco-evo model, which hopefully provides a solution to some of the issues that we'll see. So first off, what is sexual selection? I'm sure most of you are very familiar with what this is, but just in case you're not, this is a plate from Darwin's book, The Descent of Man and Selection in Relation to Sex, where he first outlined his theory of sexual selection. Sexual selection is the process whereby traits are selected because they improve an organism's ability to produce offspring, usually by improving its ability to compete for fertilization opportunities. And sexual selection happens in a variety of ways. So first of all, we can have female choice, which is where usually males display some kind of extravagant trait or loud trait or shiny trait for females, and females then decide to mate with males on the basis of their expression of that trait. We also have intrasexual contests. So again, this is usually males compete with other conspecifics in order to gain access to females, and quite often that competition involves the expression of extravagant traits or weapons, which are in some ways at least similar to the extravagant traits that are used in female choice. And then finally, something I'm not going to talk about, but which I need to mention, is post-copulatory sexual selection. So even after organisms have mated, sexual selection can still occur via means like sperm competition and post-copulatory female choice. But we're not going to talk about that, but it's the third great mechanism of sexual selection. And since we're in Liverpool, where Jeff Parker used to work, it's worth mentioning. Okay, so that's all very well and good. That's a nice piece of natural history, but what's this got to do with climate change and things like extinction and adaptation? Well, there's quite a lot of interest now in the role of sexual selection in determining sort of more macroevolutionary processes. And some of you may, for example, be familiar with ideas like the role of sexual selection in controlling speciation in some species. And sexual selection can also cause problems. So this is one of my favourite trilobites, because I have a favourite trilobite. This is Wallicerops trifurcatus from the Devonian of Morocco, one of those fantastic spiny trilobites that they get in Morocco, almost all of which are prepared by professional preparators and then sold to the private market, and hardly any of which end up in museums. Wallicerops was very, very probably sexually selected, and this thing sticking out its nose appears to be some kind of sexually selected ornament. And you'll notice that Wallicerops is extinct. Yeah? So sexually selected ornaments are costly. Yep, simply growing them and carrying them around imposes a cost on the bearer. If any of you have seen, I don't know, a widow bird in its mating plumage actually trying to fly, you'll have seen the kind of impact that sexual selection can have on an organism's sort of general fitness. Males that compete with other males can pay very serious costs. In some cases, they can get killed. But even if they don't get killed, the cost of competing can be very, very high. Females in sexually selected systems often pay a cost of being choosy, and in some cases they can suffer harassment and other horrible things like traumatic insemination, which we're not going to talk about. So overall, sexual selection should reduce population mean fitness and increase the risk of extinction. And there's some data to support this. So, introduce birds in New Zealand. the more dimorphic the birds are, the less likely the introduction was to succeed. Doherty, looking at bird communities in the USA, found that more dimorphic species had a higher extinction rate, and in fact a higher turnover rate because they would then reinvade where they'd previously gone extinct. And more recently, Bro-Jorgensen, looking at data from bovids, found that bovids with relatively big horns, this is quite indirect, but it's still worth mentioning, bovids with relatively big horns are more likely to be IUCN red listed. So there's some data to support this. I should also mention Ted Morrow and Claudia Frick looked at 1,007 species of mammal and found no effect of sexual selection on extinction risk. But there's still some data. This is a figure from the Doherty paper. It's kind of hard to work out what's going on, so I'll tell you. Anything that's green, like that bit there, is somewhere where sexually monomorphic birds have got a higher extinction rate than sexually dimorphic birds. And that's the mean value, and that is the lower bound of the 95% confidence interval. So what you can see is that pretty much anywhere in the US, sexually dimorphic birds have got higher local extinction rates than sexually monomorphic birds. The obvious exception being Texas. You might want to try and work out why that is. I wouldn't care to speculate, particularly at the moment. Okay, so sexual selection, bad sexually selected species going to go extinct. Here's the alternative. This is a male Teleopsis dalmani, a stalk-eyed fly, sitting on a root here by the Bellelong River in Brunei. And that's the male, and he's got these big eye stalks. And these females are all sitting on his root hair. And as the sun rises in the morning, he's going to be quite a busy boy. And he's going to produce a lot of offspring. And eye span in diopsids is condition dependent. The males are signalling their overall health by using their eye stalks. You've got a lot of lab data that shows this. And these females that mate with this male, they are acquiring good genes for their offspring by doing so. Okay? So males that have high quality genomes are in good condition. They signal that, in this case with their eye stalks. Females preferentially mate with them. And so those good genes spread really rapidly in the population. You get reproductive skew biased towards these males with the good genes. And so if the environment changes, particularly males that just happen to be suited to that environment get lots of matings, and their offspring are going to be more successful. So sexual selection leads to increased population mean fitness. And there's evidence to support this as well. And I'm going to go through this a bit quickly because I've just had the 5 sign waved at me. So this is data from a study published last year in Nature by Lumley. These are lines of tribolium which have been exposed to female biased or male biased sex ratios. So the male biased sex ratios have got higher sexual selection. They've been bred in this for about 60 generations. This is what happens if you inbreed them. And the males and the lines that have been exposed to high sexual selection don't go extinct from inbreeding anywhere near as fast as the ones that have not been exposed to sexual selection. And here's another data set, and I'm not going to talk about that because it's mine. I'm just going to apologise for the use of the ggplot default colours, which are quite horrible. So we've got a problem. We've got some data which says sexual selection bad, organisms that are strongly sexually selected are likely to go extinct. We've got some data which suggests the opposite, and the organisms which are strongly sexually selected are going to do better in changing environments. So we tried to address this by using an individual-based eco-evo model. All the models used so far to investigate this have basically been quantitative genetics models, and they haven't taken things like demography into account. We haven't had feedback between demography and the evolutionary process. So we built an individual-based model to look at this, and we can use that to look at the relationship between sexual selection, population size, fecundity, the kind of environmental change, and whether or not the population survives. So we've got a single important environmental factor varying on a continuous axis. It can be whatever you want it to be, pH, temperature, something like that. Each individual is more or less well adapted to this, and if they're not well adapted to it, they find themselves in poor condition. We allow the extent of their adaptation to the environment, we allow the amount of sexual display, and we allow the female preference to evolve in the model. And we control the opportunity for sexual selection by changing the number of males that a female assesses before she makes a decision about mating. So if the female only assesses a single male, the whole thing collapses back to random mating with no sexual selection. If she assesses 10 males and chooses the one with the biggest display, then you've got a strongly sexually selected system. And our males are paying a survival cost if they grow a big display. They're more likely to die if they've got a big display. So we built this individual-based model, and this is the kind of output you get from it. I apologize for the yucky colors. They are, I'm told, by a referee who was quite insistent on this, they are red-green colorblind friendly. So that's the reason we've got them. This is what happens if you have a step environmental change. The orange line is the environmental variable. And you can see that in this case, this is the population size. This is total population size. That's females, that's males. You can see the population survives that step change. The size of the display gets small and then catches up again. This is what we see with random environmental change. And again, you can see the size of the display tracking the changes in the environment, that orange line. And here you can see very clearly that we've got a strongly skewed sex ratio. And this is what we see in sexually selected populations in the wild. So things like lions, soay sheep, red deer have strong sex biases. And this is what we see under directional environmental change. So the orange line is the environment. Display is quite low, but the population, that's the population's adaptation, and it's able to track that change. So that's all very well. We then did 43,200 simulations on the high performance cluster at Queen Mary. And this heat map shows us the effect of sexual selection. So black is extinct, white is all simulations survived, and different shades of grey are for when some simulations survived and some didn't. And this is for 1,000 time steps per simulation. Two things that we could notice. Wasn't me. I'm going to just stand really still and not wave. Two things we can notice. Firstly, when we've got small populations of around 50 to 100 animals, then sexual selection makes them more likely to go extinct. Second thing is when we have big populations, sexual selection makes them less likely to go extinct. Yep, so this is the strength of sexual selection. It's the size of this is the maximum size of the population, and we've got 4 different kinds of environmental change. And you can see when we've got this directional environmental change, sexual selection rescues populations which would otherwise go extinct under directional environmental change. I'm not going to talk about that. It's basically saying what I've just said. And so, how can we explain what's going on? Well, what we can see is that the effect of sexual selection is depending on a variety of variables, and it can both have negative and positive impacts on the populations where it's operating. When the populations are large and the environment is changing, sexual selection gives us faster adaptation and a reduced probability of extinction. When populations are small, sexual selection leads to greater risk of extinction, and this is because of increased susceptibility to demographic stochasticity when these populations are small. And if we think back to some of the data we saw, those studies of introductions of birds into New Zealand that found negative impacts of sexual selection were based on very, very small populations that were being introduced. So hopefully this more holistic eco-evo approach ties together these conflicting sets of data, some from the lab and some from the field, and gives us a better understanding of the complexity of the interaction between sexual selection and extinction risk. Finally, just want to say thank you to a few people. Carlos, who actually wrote the code. John, who did those experiments I didn't talk about. Queen Mary IT support, who didn't shout at me when I wedged the high performance cluster. and the 3 guys who reviewed this for JE, who were absolutely fantastic. Really some of the best reviews I've ever had. Thanks if any of you are in the room. And that's it. Thank you.
So our next speaker is Anna. Who's going to talk to us about the technical acclimation on
Okay, so as we all know, there are 3 potential responses that an organism can show in order to survive in a changing climate. So we've got acclimatization, which is generally, I've classed it here as a change in the phenotype without any underlying change in the genotype. We've got adaptation, which is a genetic change that we'll see over many generations, and then migration, where things are moving to remain within their climate envelope. And these often have been shown that in the short term, acclimatization is often the most important for species survival. If they can respond rapidly, they can respond within a generation or 2 to actually the environment that they're experiencing. But what we're wondering is there a limit to this acclimatization? And this is potentially what we're seeing in the literature. And then adaptation is becoming more important for long-term survival, where natural selection can actually act on novel genotypes in order to adapt to the environment that they're now experiencing. But in the wild, it can be very difficult to actually tell the difference between what's happening, whether it is adaptation, whether it is an underlying genetic change or not. And actually, when you're potentially looking at long-term data sets or you're just doing observational studies, the functional phenotype that you're seeing is not actually changing. It's just the mechanism by which it's occurring that is changing. So in acclimatization, it can be generally by gene expression level, energy allocation, and that can see a change in phenotype. Whereas when you're talking about adaptation, you're talking about natural selection and actually a change in the frequency of the alleles for certain traits. So if you have lots of lovely long-term data along with quantitative genetic information, maybe you can pull those 2 apart. But often we have neither of those things. And actually the pace at which climate change is proceeding means that it's important to be able to assess this in other ways. So there's multiple ways to do this. You can look at population level variation. So populations that experience different environments, temperature specifically in this case, within their current environmental range. So you can look at altitude, you can look at depth, or for this study I'm going to talk about a latitudinal study that we did. We chose to focus on a trait that is very important for responding to temperature in the environment, and this is actually in terms of membrane fluidity. So obviously membranes are very important for a whole host of functions in organisms, and what you find is that this is the ideal state, the liquid crystalline phase, and this is the physiological temperature at which membranes are able to function. But what you find at low temperature is that actually they become rigid, they form a gel phase, and they're no longer able to function for the species, for the individual. And how we can measure this is actually to look at the unsaturation index, because one of the ways that species regulate their membrane fluidity is actually by changing the double bonds, changing the unsaturation of their membrane phospholipids. So if unsaturation index goes up as temperature goes down, then that's cause to believe that homeoviscous adaptation is going on. And homeoviscous adaptation is the adaptation that allows individuals to maintain their membrane function regardless of what temperature that they're experiencing. So we looked at this in a reef-building worm. It's called the honeycomb worm, Sabellaria alveolata. It has a latitudinal range that covers from Scotland down to Morocco, and it forms these very large reefs. They can actually be over 6 foot in height and cover a number of hectares within a site. They're very important for biodiversity in that they actually support much higher biodiversity than the surrounding areas. Each beautiful worm like this can construct its own tube out of sand, and they are gregarious. So they have a dispersing larval stage, and the larvae will choose to settle where there are other reef. And they can form these huge structures. And what's also interesting about littoral species is that they're actually experiencing a very high temperature fluctuation. And you can see the red line here shows the intertidal zone. And they can experience temperature differences between high and low tide of up to 20 degrees C. So they're actually experiencing these really high temperature fluctuations on a daily basis, making them very interesting to look at. So what we did for this paper was we sampled from 5 reefs, the most northerly known reef in Scotland down to Morocco, where the most southerly known reefs are there. And we actually collected coarse samples. So we sampled at about the time of the breeding season. They do have 2 peak breeding seasons in the year, and larvae can be found in the water column throughout the year. But we sampled at one of these and actually brought reef cores back to the laboratory, so sampled them alive. What we then did was we exposed them to a stepwise increase in temperature. So we started off at 15 degrees C and sampled them at the end of 4 weeks. Then we increased it to 20 degrees C, sampled straight away. As soon as the temperature had got to 20 degrees C, again waited 3 weeks and sampled again, and then did it going up to 25 degrees C. And these temperatures were actually chosen because if you actually look at the mean sea surface temperatures for these sites, it ensures that all of the sites are under stress at some point, and they are actually ecologically relevant. And what I want to draw your attention to in these temperatures is that actually here they're listed from most northerly to most southerly population. And you can see that actually the temperature pattern reverses itself in the Iberian Peninsula. So because of the upwelling around Portugal, you actually see that it's colder than the more northerly population in the south of France, in the Bay of Biscay. And this gives us a really nice opportunity to actually pull apart whether it's temperature that's having an influence on what we see in terms of the phenotype of the individuals, other than other things that are related to latitude. So we had a look at the lipid composition of the individuals from the different sites at each of the temperature and time points. So we can see here there's different expressions in the heat map, different expression of lipids depending on the time point. But what's really interesting about this figure is that actually it shows that here the most similar response was seen with the English Channel and the Irish Sea, so the 2 coldest sites. And what we see as well is that actually here the 2 warmest sites, so as I said, it's not latitudinally ordered, the 2 warmest sites at Bay of Biscay in North Africa clustered together. And then the Iberian Peninsula is seen as being intermediate. So already we're seeing some evidence of local adaptation to temperature. If we look at the unsaturation index that I told you is linked to this homeoviscous adaptation, we're seeing that there is a decrease in unsaturation index because there was actually a significant effect of site and time on the expression on unsaturation index, but not their interaction. And we see that it's decreasing. So we're seeing there's evidence of acclimation. But then at 25 degrees C at the longer term, we actually see that it's increasing again. So there's something happening there that is not indicative of a homeoviscous adaptation. And here we can also see that the sites here have now been ordered according to their temperature, so from left to right is coldest to warmest. And again, we see that in fact the 2 coldest sites have the highest unsaturation index, which is what you would expect. It's what you tend to see when they are adapted in these terms. And then at the hottest sites are similar as well, with Portugal again being intermediate. So there we're seeing some evidence of both acclimation and adaptation. When we actually pull this apart, what's contributing to unsaturation index, 25N-3 is actually the lipid that is contributing most to the change in unsaturation index that we're seeing. And there is a significant interaction between site and time here. And you can see here, again, the warmest sites really show a very strong change in this, while the colder sites, you're struggling to see really a pattern of change in the Scottish site. But again, they're having this strong drop in 25N-3, and then in fact, in terms of significance, there's no change between 25 degrees C and short or long term. So it must be something else that's causing this increase in the unsaturation index. So if we pull it apart a bit more, the 24N-6 is a lipid that's been very strongly associated to stress. And this was, in fact, in terms of the order of what contributed to the change in unsaturation index, it was much lower than 25N-3, but it was the next most important lipid. And we can see here that, in fact, there's not really any change between 15 degrees and 25 at short term, and then suddenly there's a large increase at 25 degrees C in the long term. So we're seeing that there might be a stress response occurring in terms of acclimation. And it's interesting to note at this point, actually, that we also recorded presence within the reef as a proxy for mortality. So you can kind of look down on the reef from above and see whether each tube is occupied. So we would measure that at multiple time points. And the only reef that we found that was decreasing was the one from the south of France. So although they're showing a stress response, they're potentially showing a limit to homeoviscous adaptation. At this point, it's not actually causing mortality. So it's not reached their thermal tolerance limit for survival anyway, but potentially for functionality. And the fact that just one site shows it, it actually has in dimethyl acetyl, one of the other lipids, this particular population is very low compared to the others. So we're wondering if that's actually some sort of health index for the reef. Potentially it's just a more unhealthy reef. So what we're finding here is, in conclusion, is that Sabellaria were able to acclimate to higher temperatures, but actually this had a limit. There was local adaptation to temperature observed between sites, but at long-term 25 degrees C, a stress response was observed across all sites, regardless of their local adaptation, suggesting that acclimation might be failing. So we're potentially saying that actually the most southerly populations are close to their upper thermal tolerance limits, and certainly southern, meaning because these populations are all very, particularly Morocco, they already experience quite high temperatures. With climate change, they're potentially at risk of local extinctions due to that. And I'm not going to talk about this too much, but I'm also doing some work on population genomics, so looking at neutral and adaptive differences between them, relating that to local temperatures recorded at each site, as well as some dispersal modeling with some collaborators in Plymouth. So if anyone is interested in talking about that, then do come and see me after the talk. But thank you to everyone who was involved in this project. It has turned into a very large collaborative project with lots of elements, and thank you very much for listening. They would, because, well, we ramped up the temperature over a course of a week. So that was the timing we did it. But as I said, actually, because they're intertidal species, they potentially could experience it actually on a daily basis. So actually, if you think about the ecology of the species, the fact that they are able to acclimatize over the short term actually makes sense, with what they experienced in the wild, because they could experience those temperature fluctuations in a day. And I think when actually you're increasing the mean temperature over a longer period of time, maybe more indicative of what you might see in a changing climate, that's when they really start to struggle. This one there.
Just to see whether you thought testing on adults was the whole. I mean, I thought it very well, but do you have any idea whether the juvenile status or the
Well, that's an excellent question, because that is also some data I have that I'm in the course of analysing, so thank you very much for that. We are looking at that at the moment, I would say. It's work in progress, so I can't actually give you any results, but hopefully they will be available soon.
Thank you. So our next speaker is Pippa from the University of Oxford, and he's going to talk to you about the sensitivity of global
fall over. Right. Okay, so I'm from a research group called the Oxford Long-Term Ecology Lab in the Department of Zoology in Oxford. I'd like to show you some research that we published this year, but it's a bit of a team effort. It's taken about 3 years to bring this together. And it's to do with this concept of resilience that everyone talks about in conservation and environment. So, for example, it's a really key target of the Aichi goals. We want to build ecosystem resilience. We want to ensure that ecosystems work in a way that's robust. But the difficulty with the concept of resilience is it's somewhat similar to sustainability. Everyone talks about it, and lots of people think they know what it means, and everyone talks across purposes. But it's actually quite difficult to empirically measure whether an ecosystem is resilient or not, and do so in a standard way that's comparable across space. So we were thinking about, well, what properties of ecosystems might you measure to be able to say whether or not they are resilient? And before we go into that, I want to briefly distinguish two different ways that people think about resilience. So when you've got a system, and you can have a sort of metaphor for it, like this potential well of a ball rolling around a potential surface, you can talk about engineering resilience, which is, imagine you've got a system, you perturb it to some way, so the ball rolls around the surface. How quickly does the system return to the state it was in previously prior to the perturbation? The other framing is in terms of ecological resilience, which is thinking of a potential surface with multiple states that it can be in. And the idea is how resistant is the system when a perturbation is applied to it from switching into an alternative state or where it stays in the state it's currently in. And it's very widely projected that there'll be climate changes and changes in mean climate state. And climate clearly is an important factor that can perturb ecosystems. But people often do models, things like species distribution models and the kind of things we've been talking about just now, that try to infer what kind of consequences a change in a mean climate state will have for organisms or systems. But the other thing to bear in mind is that there's also this phenomenon of climate variability. And it may not be quite so much the mean climate state as the degree of variability in the various climate factors that will affect systems and organisms. So, what we wanted to do was to use this sort of approach and ask, are there properties of systems we can actually measure that will allow us to sort of characterize where they fall with respect to this engineering resilience approach? So, to find a state variable you can measure and see under circumstances of perturbation what actually happened to systems. So in this case, you can take an example of a highly resilient system at the top where you perturb it and it comes back. On the other hand, if you have a less resilient system, it's a much flatter potential surface. If you perturb it, it's more likely to change. The other sort of theoretical observation that people have made is that systems which are close to undergoing a shift to an alternative state often experience increased variability in their state variables. So, for example, something like productivity. So, what we're doing in this study is not so much trying to measure resilience as such, but to measure this phenomenon of how sensitive ecosystems are to perturbations. So, it's a really useful tool or step towards understanding what a system would do in a new circumstance of climate change, for example, in terms of its probability of shifting to alternative states. So the concept we had was to measure a response variable, a state, in this case vegetation productivity, and to see in a time series whether or not it's highly variable or not very variable, and to measure a driving variable, for example, temperature, in a time series and see how relatively variable they are. So in this example, you've got very low perturbations in temperature and very high variations in the ecosystem state. So we'd say that's a system that's highly sensitive. In contrast, if you get lots of variation in the environmental variable, and yet the system doesn't change very much, that would be an example of a system that has low sensitivity. The trouble is that I'm saying, well, there's one environmental variable, and of course, plant communities and ecosystems respond to lots of different environmental variables, and how relatively important they are will differ across space. So people like Nemani and colleagues made a sort of theoretical, so this is not based on any data, they just hypothesized what would be the limiting factors for vegetation growth according to water availability, air temperature, and light availability. And they estimated where in the world would be most constrained. So, for example, the red areas are expected to be water limited. And what we decided to do was to try and put some empirical data to this kind of concept. So we took a time series of satellite data derived from MODIS for a 14-year period, and the response variable was at a monthly time step, a vegetation index that measures vegetation greenness. And then, rather than use weather station data, we used direct satellite retrievals from atmospheric columns of air temperature at the surface and of water availability, a metric of the ratio between potential and actual evapotranspiration, and then a measure of cloud cover, the proportion of looks which are cloudy in it each month. And the rationale for doing this is we wanted to build a model that would understand how relatively sensitive the vegetation response is according to these three different drivers, without knowing a priori which of them are relevant in any particular part of the world. So we did this rather straightforward method, which I'll try and explain to you. So we have response variables EVI, we have the three environmental variables I've just talked about. We also have a lag term for what EVI was like in the previous month. We then applied a masking procedure to not fit the model in months when there was, either in locations with negligible vegetation where there was very, very low EVI, or where it was colder than freezing. So that's a sort of growing season mask. The next thing was to calculate anomalies. So for example, in a particular February, you'd ask how different in standard scores is the vegetation greenness or the temperature from all other Februaries in the time series at that location. So that gives you standardized anomalies across all the variables. The difficulty then was that in different parts of the world, you'd expect the variables to be of different relative importances. So we did a PCA regression to find out for each pixel in the world which of these is the most relatively important. So that gives a weight for each of the three environmental variables. Then what we're able to do is to identify which months in time series were significantly associated with these variables. And then finally, that allows you to calculate these detrended variance ratios, the sort of concept I was showing in those two graphs with the squiggly lines, that account for the correct environmental variables and correct time periods. And that's what gives us this ability to calculate a vegetation sensitivity and to weight it for the appropriate environmental variables. So the outcome of this is a map of what we call the Global Vegetation Sensitivity Index. But before I get to that, a side byproduct of this research was to be able to empirically make, let's come out with stripes, empirically make a map that's very similar in concept to the Nemani map I showed you earlier, but this is based on data about what actually happened. So in this image, the blue areas are places which have been shown to have, in the last 15 years, had vegetation productivity limited by water availability. So obviously dryland areas come out quite blue. The white areas are places with negligible vegetation or ice caps. And then places like tropical rainforests often are light limited, things like the Amazon basin and Congo basin. And then up in montane areas in the Arctic, you're seeing lots of temperature limitation. Thank you. The next result was the actual map of global vegetation sensitivity that allows us to ask various questions. So which systems around the world are responding most sensitively to climate variability? So the green areas are the most, have the lowest sensitivity. We might consider them the most resilient areas. The red areas are the places where when perturbation in the relevant environmental variable came along, the vegetation suffered. It was much less green than it would have been. And so to zoom into some areas, up in the Arctic, where the reindeers are living, lots of red, high sensitivity regions because of this dependence on temperature and the variability in temperature. The boreal forest in North America, people have reported browning rather than greenings due to temperature stresses. Lots of water-limited areas around the world, particularly Australia. Tropical forests are really interesting. You get interesting patterns that vary. People have compared our results with droughts in Amazonia over this period. And it's temperature and cloud limited in these forests. We also know which of the climate variables those different systems were most sensitive to. And we can also look at that lag term, the memory effect. Where in the world did the sensitivity of vegetation depend on its antecedent state? And these tend to be water-limited areas where it's really important to have a soil moisture that gets systems through. So the summary is we've developed this new metric. It's a state variable that says where in the world is most sensitive to climate variability in terms of vegetation. It's a snapshot. What we'd like to do is there's all sorts of things we might do in the future, like extend this into a longer time series by using more retrospective satellite data. But we don't yet know what processes underlie these patterns. So there's a lot of work to be done in understanding these patterns. Is it something to do with plant traits or ecosystem traits or soils or disturbance, things like floods or droughts or fires? Another issue is whether these are long-lasting, over sort of millennial timescale properties of ecosystems and plant communities. And we'd like to, for example, look at pollen cores in pollen databases and see if anything about changes in ecosystems lines up with these patterns. So it's a real wide open question. The final thing I'd like to say is that we in the research group also build web-based decision support tools that serve these kinds of data decision makers. So we have a tool called Left about patterns of biodiversity. You come along and draw a box on a map and get a report about all sorts of things, including patterns of endangered species, species diversity, vegetation, intactness and connectivity, and also this resilience metric. So it's possible to use this for landscape planning and policy making. So that's all I have to say. Thanks very much for your interest.
Hi, I think it's great. It's really interesting technique. One question though, is the fact that something goes brown?
Highly resilient, and often they do. So, for example, the global food basket in India and China, where all these very intensive agriculture comes out, because of the way that land is managed, comes out as highly resilient, very low sensitivity, potentially because of the way people manage that system. Anyone?
So some of the areas that were red on one map were green on the other map with the month timeline. So is there
The memory effect.
so you've got parts of Australia that are green on
So this is simply saying where memory effect occurred in the models and explains any variance in the sensitivity of vegetation. So you can either, it's two independent phenomena. You can have important memory effects in systems, but that doesn't necessarily tell you whether or not it's sensitive. It's very similar to the idea of asking whether ecosystems are water limited or air temperature limited or insulation limited. It's just on what circumstances is the storage of water in the soils or the antecedent state important.
about ready to switch over.
Okay, thank you very much.
Thank you.
So we will stay a bit in the same framework of stability, so, but a different system. Yeah, that's horrible.
You've noticed Adaptation. That's it, chapter. So our next speaker is already done, and we'll have time to reach out to you.
So, as I said, we stay in this stability framework, but looking at this time an aquatic system. And as you know, aquatic system and all ecosystems that provide goods and services, such as drinking water, or also some touristic activities, but also some regulation with nutrient cycling. But as you are aware, and you are here in this global change session, there are some multiple stressors that occurs. Okay, let's go there. Shit. It's blocked. Oh, I think it's. Sorry. I can.
So using the arrows.
Yeah, but even.
Wait.
So. There are more than 2 slides. So there is multiple stressors that occurs in natural systems, such as climate change, invasive species, some pollution, and some land use change. And even if there is a lot of literature about looking at individual effect, which is already great, but I think that there is missing literature about how these multiple stressors interact together. And it's really important because if there is some interaction, they might have some really drastic effect on ecosystems, such as eutrophisation, leading to dead zones. So this morning, Danielle Polly mentioned this spreading dead zone around the world. And so the first thing that I want to do during my PhD is to look at how does a system respond to multiple pressure perturbation. And because it is not enough, we have also some extreme events, such as tornadoes or fires or floodings, that are predicted to have a higher amplitude and higher frequencies. So the second question that I will look at today is also how does a system respond to an additional post-perturbation? And so to do this, to answer to this question, I switched to the lab because it's not really ethical to manipulate the Lake of Zurich, for example, where I come from. And so I use a community of unicellular organisms, which are the ciliates, that feeds on algae and bacteria. So the whole community was about 20 species. And on that community, I applied firstly 4 different stressors. So the nutrient with a nitrogen and phosphorus ratio, a carbon enrichment, light, and temperature. And so I tested these 4 stressors at different, I did some preliminary experiments, and it's resulting to have these 2 levels. So the white is the control, and the red is the stress control. And then I applied a 5th perturbation on half of the replicates to have this density-independent mortality. And in other words, is that I replace half of the system by a fresh medium. So it resulted to have 32 treatments with replicates. And so for that experiment, I could collect some individual data, population abundance, and also some measurement of the dissolved oxygen, and it's this result that I will show you today. So if I explain to you how I did this experiment, so for the first 8 days, all of the microcosms, they were at these control levels. So here the y-axis is the percentage of dissolved oxygen, and then the red line corresponds when I applied particular stressors. Then all of these 6 replicates just follow a response. And then on the 24th day, I applied this 5th perturbation. So in black, it's the same perturbation that was previously acting, and the blue lines correspond to this replacement of the medium. And so here it's all of my data. And so how you can see, so here it's all of the control. The blue points represent the replacement treatment. The T, if there is 1 letter, it will mean that there is only 1 perturbation that occurs. And here on the top, it's when there is all of the perturbation occurred. And so because from this time series, it's not really easy to extract some information, I used the 3 ecological stability variables, which are the resistance, which is the maximum difference between the control in white and a particular microcosms, and also the resilience, which was from last talk, it was this engineering resilience, which is the rate of recovery at this day of maximum perturbation, and also the return time, which is how much time does the system need to recover from perturbation. So for the first part, how does the dissolved oxygen respond to multiple pressure perturbation? So first, with the resistance. So here, the more negative the value are, it means that the oxygen is completely depleted in the system, and it means that the system is not stable. And here in the y-axis is a number of stressors. So what we could observe is the microcosm that had the treatment of carbon enrichment, they had a much more decrease in oxygen. And this is due to the carbon that act a source for the bacteria that consume this oxygen. What we could also observe is the treatment of the light reduction, resulting to also have a decrease of oxygen in the system due to an effect on the algae. And we tested also the interaction between the stressors, but we couldn't find anyone, and that was completely actually frustrating. And another interesting result, if we look at the number of perturbations, there is still this detrimental effect of the number of stressors on the resistance. Now for the resilience, which is the rate of recovery after perturbation. So the more negative the value are, it means the rate of recovery is much faster. So the system will be more stable. And so to have this recovery, what's triggered this recovery? It means that the production of oxygen has to be higher than its consumption. And so what we could observe is, like, again, the carbon enrichment has a significant effect, and this is due to the effect of the carbon that initially affected the bacteria, and then the ciliates can grow, and there is this top-down effect from the ciliates on the oxygen. Or there is a second hypothesis, is the carbon can also be used by the algae that will produce much more oxygen. So we tested the interaction and the effect of the number of stressors, but we couldn't find any for this variable. Now for the return time. So here it's the time, the day when they recover from the perturbation. And so the lower the value are, it means the system is more stable. And what we can see, it's again this carbon enrichment has a significant effect, and this is due to the carbon. Okay. The carbon that was using by the bacteria, then the ciliate, and then the predators can increase in abundance, and that has this top-down effect compared to the oxygen. There was also an effect of the light that increased the time to recover, that's affecting the algae. And here for this variable, we could find some interaction between the stressors. So with the nutrient ratio that interact with the light and the temperature that interact with the carbon enrichment, that leading to increase the time to recover. And what we could see also is that when we increase the number of stressors, the return time increased also. So now for the second part, so with this fifth perturbation, so for the resistance, what we could see is the more negative the value are, the less oxygen is in the system, just a reminder. And so we can also see that if the system is previously disturbed by the carbon, then the resistance is much smaller. But we couldn't find also any interaction between the stressor or any effect of the number of perturbation on this variable. For the resilience, here it's simple, we couldn't find anything. And for the return time, we couldn't find any effect of one individual stressors, but we could find some interaction between the temperature and the carbon enrichment, that if the system was previously affected by these two stressors, the time to recover just increased. And also there is this three-way interactions with this indirect effect of the nutrient ratio affecting this carbon and temperature interaction. And interestingly is that if the number of perturbation increase, there is an effect on the return time. So to summarize, how does the dissolved oxygen respond to multiple press perturbations? First, we can see that there is this really large effect of this carbon enrichment that destabilize the system by decreasing the resistance and increasing the return time. But also it was a stabilizing effect by accelerating the rate of recovery. And when we discussed about this result, we were thinking about it might be due to the experimental design because we had only 2 levels that we tested, and maybe this particular stressor was too high. But actually in natural system, you will always find a larger stressors. What we could see also is that there is a detrimental effect of the number of stressors for the resistance and for the return time, even if we couldn't find interaction between the stressors for the resistance. So that might be due to a lack of statistical power for this experiment. But to do that, I should have been increasing the number of replicates, and after it's just not feasible for one person. And so for the other parts with this another post-perturbation, what we could see is that the system will take more time to recover when there is a previously highly disturbed. And on that, I would like to thank my group and my co-author, and of course you, for your attention. Thank you. Yes.
Is there a reason why you monitor them like as a factor, not continuously, like in quantity of chemical you gave to the experimental setup? Because it looked like, how would the pattern look like if you measure it on a continuous scale rather than 1, 2, 3, 4, 5?
I don't get, because the stressor, they have just 2 levels, so we cannot have any continuous effect. I don't know if it's your question.
Do you give different quantities of the stressor, or is it the same? Wouldn't that be?
No, it's the same. It's just, yeah. But I did not measure the level of nitrogen that was still available after. Thank you.
Thank you, Aurélie. We'll move on to the next. Our next speaker is Thomas Parker, who's going to leave us on thinking about philology and ecological adaptation.
Okay. Thanks very much for coming along. My name is Tom Parker. I'm a postdoc at the Marine Biological Laboratory in Woodsall, Massachusetts. And I've been working on a project with Jim Tang, Mike Moody, and Ned Fetcher on the adaptation of a very common species in the Arctic called Eriophon vaginatum, which we think is very well locally adapted wherever it grows. So I'm going to be telling you about one subset of our project, which has been looking at the phenology of this species. So before I get going, I just wanted to acknowledge that the work that we've been doing hasn't been possible without the help and the contribution of lots of different people, collaborators, PIs, especially the 3 at the top, the 3 PIs who really got this project going. So the species we study is Eriophon vaginatum, commonly known as tussock cotton grass. It is a tussock-forming sedge, so it's made up of lots of different tillers which form these growth structures, which grow to about 150 years old. It has a circumpolar distribution, but can really be found anywhere that's cold and wet, really. So you find it very commonly in the kind of northern England or Scotland, things like that. It's a very important part of tussock tundra, so in the north of Alaska, you will find it contributing up to about 30% of net primary productivity. And it's really a structural centerpiece to the ecosystem, so all the other species kind of form themselves around these tussocks. And it's incredibly important in Alaska, where this map of tundra regions in Alaska, the yellow areas are the areas that are dominated by Eriophrum vaginatum, so in moist acidic tussock tundra. And just to kind of pile this point home, this is just a picture across the landscape at Toolik Lake where I work. And all you can see for hundreds and hundreds of miles are these tussocks. So it's a pretty homogeneous landscape in certain ways. Now, this species has been studied since the very early 1980s. Ned Fetcher, Gus Shaver, and Terry Chapin have been studying this species, and you can see a picture of a tussock there. And they saw variation as they drove up and down the whole road, which is this hundreds of miles long road that goes from north to south in Alaska. And they wanted to know whether these differences they observed in tussock morphology and growth rate, things like that, whether they were varying because of environmental controls or genetic controls. So what they did was a common garden reciprocal transplant experiment where they took tussocks from 6 different sites, which you can see on this map over here, and moved them between the different sites. And to summarize 35 years of great research into a few bullet points, what they found at each of the sites, regardless of the environment, they saw constant differences in morphology between the tussocks, in biomass accumulation, in physiology, and ultimately survival. And so they started to hypothesize that this species, because it's so well locally adapted, could suffer from adaptational lag. So because it's got low genetic variation and potentially because it's got low gene flow. So they did these manipulations purely from trying to understand whether these differences they saw were genetically controlled. But now we know that climate is changing very rapidly in the Arctic. It really brings it into focus. So the Arctic is warming faster than anywhere else in the planet. This is resulting in a number of different changes across the Arctic. One of these is increasing productivity. So as we've got warmer temperatures, we've got increasing photosynthesis and increasing biomass in plants. Another change that's happening is that the growing season is getting longer. So the main winter snowpack is melting out earlier, and it's accumulating later in the season. So we've got a longer growing season. So for a very well-adapted plant species that's been there for 10,000 years or so, this rapid change could potentially cause a problem for them. So it kind of raises a couple of important questions. Are growth rates and the phenology of Eriophrum adapted to the local conditions that they were from? And furthermore, which is a question which we can't quite answer yet, will phenology be able to track changing climate where we've got longer growing seasons in the future? So just to present a really naff schematic here, we've got populations in the south here and populations in the north, both with different lengths of growing season. So in the south, we've got a longer growing season. The hypothesis is that these tussocks, their period of growth is just as long as the growing season that they inhabit. And then the question really is, if the growing season is then longer, will they be able to extend this growing season, or are they kind of genetically inhibited? So the experiment I'm telling you about is another transplant experiment that was done in 2011 by Ned Fetcher. Ned is a guy who just likes to move tussocks. I mean, in the last couple of years, I've moved a lot of tussocks around Alaska for him. But he did this in 2011 and took tussocks from each of these 6 different sites. And so I should mention here that tussocks are incredibly good for transplanting, because what you do is you cut underneath the rhizomes with a bread knife and move them to anywhere, put them down, and they reroot. So they have a deciduous rooting system that is really, really good for this. So you can move mature individuals, which is neat for this kind of work. So we moved them to here. This is Toolik Lake Field Station in the north slope of Alaska. So it's that spot just north of the Brooks Range in Alaska. And it's an incredible place to work, and where I've been working for the last couple of years. We then warmed half of them with open-top chambers. And what we did was we tagged individual tillers on these tussocks, so we could go back to them and make repeat measurements through the growing season. So we measured their their growth rates, and then we also measured the length of senesced portion on these tillers. So, and we kind of tried to pinpoint when senescence started for these different populations. So to jump into the results, on the x-axis, what we have here is the length of the growing season at the home sites from where these tussocks came from. On the y-axis, we have growth rates in both 2015 and 2016. The letters just correspond to where they're from, just to kind of clarify in case anyone happens to know these spots, which is, one of them is called No Name Creek, so I doubt you know them. But anyway, there's no difference in growth rate at all in either years. So they are, they're all in the same spot and they're all growing at the same rate. There's no effect of warming either. And which is interesting because they've seen differences in biomass accumulation, but it's clear that there's no differences in growth rates. So again, to look at a new graph, on the x-axis we've got the length of the growing season again, but on the y-axis there's the date at which senescence started. And what you see with the 6 different populations growing in the common garden is that the 2 populations from furthest north, which have the shortest growing season, senesce earliest in the growing season, or so senesce earliest when they're in this common garden environment. And this is consistent in both years of measurement, 2015 and 2016. So another thing that we can measure on these tillers, or estimate, is the date at which they reach their peak biomass. So this takes into account the entire tiller as opposed to just when we see the first sign of senescence. And what we see again on the x-axis, the length of the growing season at the home site, and then on the y-axis, the date at which they reach this peak biomass when in the common garden. And what you see again is this positive correlation between the length of the growing season at the home site and the date at which they reach this peak biomass. So it seems like phenology it seems like there is a link between this environment that they've been adapted to and phenological stages that they seem to exhibit in a common garden. So some of you may have noticed that there were some differences in these kind of senescence and phenology stages between 2015 and 2016. So generally 2016 was later in the year. And that's consistent for both these 2 metrics, and it's consistent across most of the different populations. The interpopulation differences stay, but there seems to be a general shift to a later senescence. So we kind of dug into the meteorological records at Tulip Lake to try and kind of work out what was going on. It should have been pretty obvious, actually, because just ignore the photos just for a second. If I just draw your attention to this figure on the right, what you see is air temperature in the 2 different years. Let me try and use this thing. No, I won't use that thing. So the snow melted out at about the same time, but then in 2016 it stayed cold through May, and even into June it stayed cold, whereas in 2016 it's kind of stayed warm through both months. And what we think happened was that this cold period that we got in 2016 delayed the growth of, or the start of growth. And because we think that the period of time that they grow for is genetically constrained, because it started late, it finished late. So that's what we think kind of caused this difference between the 2 different years. And you can clearly see, that's a very dramatic example, but you can clearly see that 2016 was colder at the beginning of the season. So just to wrap this up, we do see ecotypic differences in phenology in this species, which is a first in the observation of this species. And we think that this could be due to a selection pressure at their home site, which is the length of the growing season. Growth rates in phenology aren't responsive to the warming treatment, again suggesting that this is under some kind of genetic control. And also that phenology can be shifted by early season conditions, but the length of the growing season the length of the period that they grow for, it may be under genetic control, so selected for. I'll probably just skip past this fairly naff schematic, and thank you all for listening.
Delighted somebody's study of sketches. Very interesting thought.
Oh,
You mentioned potentially there might be differences. Are there any plans that to go on and look at things next?
yes. Yeah, thanks for asking. So yes, our collaborator on the project, Mike Moody, is a population geneticist, and he has already identified that these populations are in fact genetically distinct. Being an ecosystems ecologist, I can't go into the details of the methodology and what makes something really distinct from another, but he has identified them as kind of distinct from each other, and he's kind of saying that there's probably very low gene flow between these populations. And he's also trying to look at the transcriptome as well, so measurements of these different populations at the same time, but trying to measure which genes are actually being expressed at that same time. To my knowledge, there are no tussocks. To my knowledge, I could be wrong. I know there is. I think there's some plant life down there, but Oh, okay. Yeah, I'm not sure I could answer that. Yeah, because I'm not well versed. Yeah.
Did you say how much warmer the plants were?
With the open-top chambers. Yeah, they warm by approximately 1.5 degrees on a sunny day. So that's not actually a very large warming compared with kind of daily variation, but it is a kind of consistent treatment. So the accumulation of what we use thawing degree days, which is like the accumulation of temperature above 0 degrees C. That is quite a large treatment if you take into account all the days of warming, but it's 1.5 degrees is something that we've already experienced in the Arctic. But yeah.
What do you know about the history since the last glaciation? Where were these species or this species in the last glaciation?
That's a good question. It's something I wish one of our collaborators was here to answer, because he's very interested in the glacial history of Alaska and how long these different plants have had to adapt, or to their local conditions, and that I'm afraid I don't know.
We'll have to finish the questions then, but thank you so much.
Thank you.
So our next speaker is Rangifer. He's going to talk to us about genetics of his over 17-year experiments of simulated climate change.
Thanks very much, Pippa. Okay, so we'll start here. I'll frame the questions by introducing the study system. This is Buxton Climate Change Impacts Lab. It's a long-term manipulation of the climate over an intact grassland ecosystem. So the plant community here is a species-rich grassland, and it's developed over carboniferous limestone. So this is a calcareous grassland. The figure on the right here just shows the climate treatments that are being applied at Buxton. These have been applied. Next year, the experiment will be in its 24th year. So this is one of the longest manipulations of climate over any intact ecosystem. So the treatments include a drought treatment, and let's see if we can get this to work. Yeah, so these white squares here, they're rain shelters. So they apply a 2-month drought treatment in July and August of each year. So that's a really significant drought treatment that's applied on an annual basis. And we have a control system here that senses when it's raining during those 2 months and sends the shelters over the plots during a rain event, and then they retreat again when the rain stops. So hopefully we're not getting too many additional shelter effects there. There's also a winter warming treatment, which runs from November until April each year. And here, soil heating cables are used to warm the temperature within the soil and at the soil surface by 3 degrees centigrade relative to the ambient conditions. Other treatments at the site include watering, 20% above long-term average, and then there are factorial combinations, heat and drought, and heat and additional water. So it's a really neat resource to investigate responses to climate change in an intact ecosystem. We've got no problems here with the ultimate attribution of effects. They should be down to the climate treatments themselves. The little cartoon here just shows a schematic of one of the plots. This is a 3 by 3 meter, the plots are 3 by 3 meters, just so you get an idea of their scale. And in fact, within these plots, because these grasslands are so kind of tight, if you like, you've got up to 500 individuals of each species within each of these plots. So you get a genuine population within each of the plots. There are 5 replicates of each of the treatments arranged in blocks across the site here. So for the first 13 years of this climate manipulation, the story was one of resistance to climate change. This graph here on the left-hand side just shows how the abundance of different functional types of plants have changed over time on the x-axis with some of these climate treatments. So here we have the relative abundance of these functional types. So we've got, in fact, forbs, grasses, sedges, and shrubs. And you can see that in nearly all of these, the width of these bands kind of stays the same through time. So we don't have any real extinctions of species from the plots across this period either. So that showed essentially that the system seems to be quite resistant to change. In the last 13 years, we've detected some more fine-scale changes in the abundance of individual species. So the plot here just shows the response to warming of some of the most common plant species at Buxton. So a positive response here means a change in abundance, a positive change in abundance relative to control, and a lower score here represents a decrease in abundance relative to control. And in fact, the change in abundance, the climate response here, is correlated with a functional trait of these plant species. So each point represents a different plant species. We can measure this trait under control conditions and use that as a predictor of how well the plant does under warming. So the idea here is that because it's winter warming, the growing season's extended, and therefore the plants need to be able to essentially keep growing to outcompete their neighbour and remain competitive in such an environment. The point I want you to take home from this, though, is that although we have this relationship, the explanatory power is relatively low, and so the question is kind of what are some of these points doing so far above the line. So we have, in conclusion here, relatively little change over time, and what change we do have isn't well explained by the potential fitness of the species. So what I became interested in then was whether there's the scope for evolution or genetic adaptation within species for allowing this resistance to climate change. And so that introduces the aims of this talk. I've spent the last, 6 years studying this, so trying to work out how plant phenotypes have changed and whether plants are adapting to climate change, whether that's a part of this really interesting resistance to simulated climate change we can see at Buxton. So first of all, I'm going to run over some of the phenotypic responses I've documented, but then we'll get on to the main kind of point of this presentation. Is there a genomic signature, a true genetic signature to natural selection driven by these climate change treatments? If so, which genes are involved, and can we say that there are particular gene functions that are important for adaptation? Here are the 2 study species we'll be looking at. Festuca ovina, that's tetraploid. Notice that it's got also a high a very large genome size. That's a picogram genome size. And that's relevant to the methods that will be used. And we also have Lotus corniculatus, a legume. So Festuca is the most abundant species at this site. It's the matrix within which all the other species are growing and having to perform. So all of this work then depends on a series of clonal lines that I collected from Buxton back in 2010. The 17 years in the title of the presentation refers to the 17 years from the beginning of the experiment until 2010, when I removed the plants from control plots and from drought-treated plots, and I brought them back to here, which is Nest Gardens. So they were grown here in a common garden environment, and I've used them for various experiments since then. Here are the two study species, Festuca and Lotus. So we have a population of plants maintained as clonal lines that were collected from control plots, and a population that was also brought back from the drought plots for these, well, in fact, these 4 species, but we're only here today about 2 of them. Okay, so here we're going to look at some of the phenotypic responses in these 2 species. These are the phenotypes of F0 plants, so the clonal lines that were brought back from the field. So here for Festuca, we can see inflorescences is essentially reproductive. It's the amount of flower heads that the plants produce. This red here is the population from the drought plots. So the population from the drought plots produce significantly fewer seed inflorescences than those from the control plots. The pattern is the opposite in Lotus, which is an important nectar resource for bees. So these plants have adapted by essentially producing a greater number of flower heads. So here, again, specific leaf area. We have a decrease in specific leaf area in Festuca, and this reflects the pattern we see between plant species. So plants from more arid environments, in fact, tend to have lower specific leaf area. And here, again, is Lotus corniculatus, showing that this population under drought flowers later. We also have these phenotypes being maintained into the F1 generation. So here is germination latency data, so plants from the drought plots germinating later for Festuca ovina. And for those who were here earlier, the plant botanical equivalent of climate change shrinking reindeer, not as festive, but just as interesting, I hope. So a biomass response here, the drought plants producing significantly less biomass. So essentially what I've been doing is some genomics work since then. I've sequenced the transcriptome of 2 of these species, Festuca and Lotus, collecting tissue from different parts of the plant, and then exposing that to next generation sequencing. I've assembled the gene transcripts, and I've annotated them with their functions. I've also developed a sequence capture assay, so I can use probes to get at that gene complement of each of these species. So essentially the rationale is we have some genomic DNA, we mix it with some probes, and then we pull out the gene sequences of these plants, and then we can sequence them and study the response to climatic selection. This is the design. So essentially what I've done is I've gone to different environments at Buxton, control plots with deep soil, control plots with shallow soil, and so on. And I've taken populations of plants, extracted their DNA, and pooled it. So that gives us 4 samples. And then I've used a sequence capture assay to pull out the gene sequences from these 4 samples. And I've sequenced it on next generation sequences. And then we have some genic sequences that we can look for genetic differentiation within. And here is one of the results for Festuca ovina. What we have here is a plot of single nucleotide variants. So there's more than 150,000 single nucleotide variants that were identified. The y-axis measures FST. That's the difference in allele frequency between the drought-treated and control plots. And low values here mean that the allele frequencies are the same in these 2 environments. And high values mean that they're very different. So the red points here represent statistically significant outliers, so alleles and loci that we expect should be under climatic selection. So we should expect actually this low baseline for most loci because of gene flow between the plots. Okay, so at the most stringent level, we have this response to selection. So here's the functional response. This depends on a kind of slightly lighter touch. We're accepting some false positives using a lower threshold for FSD here. And what I've done is use the annotation to see if particular gene functions are being used to mount the adaptive response. In Lotus corniculatus, there are very few overrepresented gene functions, or those that could be disproportionately involved in adaptation. Okay, so we've got these heme metabolic processes here. They're thought to be important in drought resistance and stress signaling. The surprise comes in Buxton, in Fastigia ovina, where we see these responses associated with the immune system of plants. So this looks like it might be an indirect response to the environment. So I might have ignored that were it not for the fact that in a separate system where there's natural moisture variation, I've identified exactly the same functional response. So we have discovered this genomic signature of climatic selection at Buxton. It seems to be operating in different ways in different species with different classes of genes. And the results with Fastigia seem to suggest that there could be indirect responses to selection, so that perhaps rather than climate change imposing direct selection on plants, that could be imposed indirectly by other partners in the ecosystem. And there I'll stop. Yeah, so I targeted the whole transcriptome, but because of the way these things work, the effective coverage of the probes was about 2/3 of the transcriptome. So it was quite a big sequence capture. Yeah. Yep. That's a really big question. That's something I'm trying to kind of follow up. So you will have noticed that there's no obvious connection than the phenotypic differentiation that was observed and the sorts of functions that were coming up for Fastigia ovina. So the next big thing, I suppose, is to try and put these together and to understand how that's working. I mean, it's fascinating. Yeah.
Seconds and counting. I will do a quick announcement if the chair says I may. Hopefully, a lot of you, all of you, are interested in climate change and ecology. So therefore, you should be interested in tonight's Climate Change Ecology Group social event, 6:50 in reception to walk over to the Pump House, which is pub in an old building in the Albert Dock, or 7 o'clock there. Places are limited, but there is free food. You have to buy your own drinks, though. Speak to Pippa or speak to me afterwards. Right, okay, resilience then, building resilience to climate change. I'm going to get my acknowledgement in at the start, just one really to everyone involved with the UK Environmental Change Network, the data from which I draw on extensively. So resilience, it pops up everywhere today in the BES meeting. I'm really glad you're here and not listening to Georgina Mace right now, so thank you. But you could have gone and heard her views on resilience, and you'll never know if that would have been a better choice. But anyway, resilience does pop up everywhere. Right, who knows what's going on here? Interaction, end of the day. Absolutely nothing at all. No, this is a landmark. This is some chap, Johnson, I think his name is, signing up for the UK to the Paris Agreement on climate change, roughly a year old. And writ large across that is resilience. Now, we think about it all in the Western world in terms of building
or reducing emissions and so on. But actually, from the developing countries, building resilience is a really important topic. And actually, it's a really important topic for ecology and conservation as well. And you can find the R word writ throughout things like the Lawton Review, the National Adaptation Programme for Climate Change, and my own organisation, Natural England, loves it dearly as well. But it probably hasn't escaped many of your notices that resilience can be quite a contested topic and quite a difficult topic. But I think it's a really important one, and I would say to anyone who's going to get bogged down on definitions, if you really care about making a difference in the real world, we have to get real about this. This is one definition. We have to work with what's out there, the language that people are using, and resilience is an important term that we can work with. This is the Intergovernmental Panel on Climate Change's take on resilience. The ability of a social or ecological system to absorb disturbances while retaining the same basic structure and ways of functioning. The capacities for self-organisation and the capacity to adapt to stress and change. It's conceptually useful, and I really mean that. It really is useful. It's a word that people do relate to outside our little scientific world. But let's face it, it's quite difficult to measure. And if we can't measure it, then how do we know if we've achieved it? So there's a tension there. And my attitude really is, well, we have to live with that, and we have to work within that. This is a paper that colleagues and I wrote a few years ago, and this was the conclusion. For practical purposes, let's use a pragmatic approach. Accept resilience is a broad term, but ensure when we do use it that we clearly explain what we're talking about. And I was quite satisfied when we'd written that. It's always nice to get a paper in a good BES journal. But moving on, life moves on. Well, how do we measure it? How do we actually quantify it? So this is the latest sort of installment in my thinking, and it's actually going down from the big scale, which I work out a lot of the time, into a more detailed study. And I want to pick up on another concept related to resilience, which I'm calling stability. We can debate in the bar tonight whether or not that's the right language, but what I'm trying to do is find something that we can measure, and I'd argue that there are things around stability that are easier to measure than resilience, and therefore useful. What do I mean by stability? This is my definition. The extent to which a community, and I'm talking for the moment about biological communities, particularly plant communities, the extent to which a community's composition, the balance between species, or properties remain the same over time. And so broadly speaking, we'd expect that stability, that consistency to increase with the resilience of a community. A more resilient community will be more stable. But it's also obviously affected by the pressure, the disturbance, for example, that a community is put under. So it's a realized property rather than a fundamental property of, oh dear, that wasn't good, was it? Oh, there we go, it's back again. Rather than some sort of fundamental property of the system. But that's advantageous, because I think that stability may be an indicator of resilience, assuming, of course, that the pressure on those different communities is not radically different. And so I've gone on to look at what happens in terms of responses to different changes in the weather at different sites across the UK. The weather, yes, it does differ, we know that, between different sites, but the year-to-year trend is broadly similar across the country. So these were the questions I wanted to ask, working with colleagues, particularly Clive Bealey, my co-author, who I should have acknowledged at the start. Are some habitats more stable than others? Habitat in the sense of plant community. And is that stability related to the diversity or the functional types of species within it? And we use data from the UK Environmental Change Network, a network of ecosystem monitoring sites across the UK, which pulls in a wide sample of different British vegetation, uses a standardized methodology, For today's purposes, you don't need to know, other than we're looking at species lists and how they change year to year in different vegetation monitoring quadrats. And for at least some of the sites, in some years, we've got annual repeats. So we can pick up the way the community itself responds to changes in the weather. Throw all that data from all years and all plots and all sites into a great big analysis, a Kanoko analysis, and plot it all out as an ordination. If you're not the sort of plant ecologist or indeed community ecologist generally who uses these sort of data, tough luck, I haven't really got time to explain. But broadly speaking, it's a measure of how similar the community is in terms of its species composition. So the closer on those two axes that a point is to another one, the more similar that it is. There are fascinating things I could pull out of there, and you can see it pulls out nicely different types of vegetation. But actually, I want to go into the detail of how some of these points move around. So within that jumble, if we look in a bit more closely, this includes year-to-year data. So in this case, the different colours there represent vegetation in different years in the same plot. And you can see it moves around the ordination analysis. Communities change from year to year. How can we quantify that? Well, we can look at the variation in the two different ordination axes. And I've got along the bottom x-axis here some very broad vegetation classes, a classification derived from the Countryside Survey. It doesn't really matter too much about the detail. The important thing is, though, that on the left-hand side, there are the more disturbed vegetation types. So these are typically lowland, agricultural, more high productivity grasslands, road verges, that sort of thing, going through different types of woodland and semi-natural grasslands, through into the more upland systems, bogs and heath on the right-hand side. And if you look, the really large amount of variation, okay, how far up the x-axis it is particularly in axis 2, and it's particularly associated with the most disturbed vegetation types. So, in a nutshell, the vegetation that's least variable is that associated with cold places, fairly slow growing vegetation, bog, heath, that sort of thing. So that's interesting. Can we look at it a bit more? Well, we can do another index of stability of species composition, which we can use at a plot level. And this is a simple index showing the number of species in each pair of successive years as a percentage of the total number of species in both years combined. All that really means is, what's the second bullet point there? If it's 100%, then there's no change in species between years. If it's 0%, there's a complete turnover of one community being replaced by another. So the higher it is, the more stable it is. Plot that out for the different plots across all this wide range of different British vegetation type, and you can come up with some interesting correlations. The stability is positively associated with the number of species. More diverse communities tend to be more stable. But it's also associated with species that are typical of low nutrient habitats. So the Ellenberg N number is an indicator of the fertility of sites which are usually occupied by species. And finally, there's an association with the trait of stress tolerance in the sense developed by Philip Grime and co-workers at the University of Sheffield. The S coordinate measure on the right-hand side here shows that the more stress tolerant the average species is within a community, the more stable that community is. So, some conclusions then. First of all, just to go back up to the big scale, Resilience is an important concept for climate change adaptation, but also actually a wide range of different policy objectives. I was quite pleased that Natural England actually has resilient landscapes and seas as one of our top 3 headline objectives. But community stability, what I've been talking about in terms of developing a measure, can help us to understand something about resilience. It's not a perfect measure, no indicator ever is, it is just that. But it is measurable, and therefore it's something where we can move towards a more quantifiable approach. Upland grasslands, heath and bog communities tend to be the most stable. Disturbed lowland grasslands, the least stable. And in terms of species, the stability was associated with a high diversity, stress-tolerant species, and low-nutrient adapted species. Any of you who work on those fields, you know it's not a surprise those 2 attributes are actually quite often correlated in any case. There's further work we could do. We haven't attempted to disentangle resistance, if you like, the ability to resist change in the first place from the speed of recovery. Both of these would contribute towards stability as we've measured it, so there's a lot more work that could be done there. But I do think, and this I think is going back to the big picture, it's a hint, quite a good hint, that if we can restore some of the attributes of semi-natural communities, low nutrient tolerance, stress-tolerant species, a stable community, we can actually start to build resilience. And in the context of adapting conservation to climate change, adapting land management to climate change, then that's a really useful piece of information to know. Thanks. So what communities? Bog communities?
They are very poor in species composition.
Well, I don't think that's necessarily true, actually. If you take into account the bryophytes within them, I think they can be relatively species rich. If you compare it to some of the improved grasslands that we have in the south of England, where you may just have 2 or 3 species, they are relatively rich if you include the full range, including the bryophytes. I mean, it's a very broad-scale comparison, and I think there would be some interesting, more local-scale, more detailed comparisons one could get into, about that, comparing, let's say, a chalk grassland with, perhaps 40 species or something like that, with a bog containing 20 species. I don't know. I don't know how it breaks down if you go into a more detailed level, but at the really coarse level, I think it does actually hold.
How do you handle the fact that if you have 2 species and you lose 1, that's a big change. If you have 10 species and you lose 1, it's just 10%. So part of the stability, diversity is an arithmetic game.
Yes, I think there is some truth in that, actually, but isn't that one of the good things about a diverse system? You can afford to lose 1. On the other hand, I mean, the other thing I'd also throw in is that also very often if you do have the communities, with 30, 40 species, they may be in very low percentage cover or number of plants within it. So I think you've got to know that, if you want to look into the detail, you have to look at the specific situation, but you could also certainly conceive that there are some extremely rare ones that are on the brink the whole time. Yeah, there probably is, yeah. I think it was trying to find something that we could use at the whole country level. And I don't have a problem that there may be other measures or better measures. There are different approaches, and I think it's a fruitful field to look at more. That's a good point.
Yeah, consider there's a lot more questions to Mike, but we'll come to our social to ask them later.